Now, the next topic is prokaryotic cells. Prokaryotic cells are the cells without nuclear envelope. A cell which is bounded by a cell membrane and inside it has a cytoplasm and there is present no nuclear envelope. The genetic material present here is in the form of a small circular single chromosome is present which remains attached to the cell membrane at a point. यहाँ पे न्यूक्लियर एनवेलप नहीं है. जो भी क्रोमेटिन नेटवर्क या जेनेटिक मटेरियल है दैट इज प्रेजेंट इन द फॉर्म ऑफ अ स्मॉल सर्क्युलर single chromosome which remains most of the time attached to the cell membrane, and this is called nucleoid. The only cell organelles present in the prokaryotic cells, they are the ribosomes. बाकी जितने भी सेल ऑर्गेनेलीज हैं, एंडोप्लाज्मिक रेटिकुलम, गोल्गी apparatus, माइटोकॉन्ड्रिया, vacuoles, all the cell organelles, they are absent. In prokaryotic cells, only cell organelles present are the ribosomes, and these ribosomes are different from the found in the eukaryotic cells. These are 70S, and in eukaryotes these are 80S. In some bacteria, in addition to this nucleoid, there is present single or more than one, maybe up to 300, 200. A circular DNA is present which consists of special genes. यानी बैक्टीरिया को सर्वाइव करने के लिए जो भी जीन्स चाहिए, जो भी फंक्शन है वो तो इससे कंट्रोल हो जाता है, लेकिन ये जो एडिशनल सेगमेंट है डीएनए का प्रेजेंट, सर्कुलर डीएनए का, विच इज कॉल्ड episome or plasmid, it is seen in some of the bacteria and it carries certain special genes, और वो special genes जो हैं they can perform special functions in those bacteria only. For example, they may have a gene for antibiotic resistance. तो जिस बैक्टीरिया में ये जीन होगा that will become resistant to the antibiotics. The cell membrane may have infolds at places, and these are called mesosomes. These are called mesosomes. Now, if the bacteria, it is surrounded by a cell envelope, and cell envelope consists of 3 layers. The innermost is the cell membrane. Outside the cell membrane is present cell wall, and there is a third layer, outermost layer, made up of glycocalyx. Outermost layer is of which is called, it is called glycocalyx. And the third layer, outermost layer, is called glycocalyx. Depending upon the nature of glycocalyx, it can be, glycocalyx can be a loose sheet which is called the slime layer. अगर यह glycocalyx एक loose sheet है, then it is called slime layer, and if this glycocalyx layer is thick and hard, then it is called capsule. यानी अगर बैक्टीरिया की outer most layer it is thin, loose and slimy, then it is called slime layer. But this glycocalyx layer, it can be thick and hard, then it is known as the capsule. Now, the bacteria, they can be gram positive and gram negative. Means the different staining procedures, by applying a different staining procedure, means the bacteria which can take up a gram stain, they are called gram positive, and those which are not able to take up gram stain, they are called gram negative bacteria. So the gram positive and gram negative bacteria, this technique was developed by Christian Gram, and the stain used to dye the bacteria, that is called gram stain. In this, कैसे हम पता लगाएंगे? The bacteria, they are treated with weak alkaline solution of crystal violet. When bacteria, they are treated with a blue colored dye, crystal violet, or the crystal violet, these are dissolved in weak alkaline solution, तो all bacteria they turn blue. यानी कि बैक्टीरिया को जब हम ट्रीट करते हैं विद अ वीक अल्कलाइन सॉल्यूशन ऑफ क्रिस्टल वॉयलेट, तो सारे बैक्टीरिया दे बिकम ब्लू इन कलर. नाउ दीज ब्लू बैक्टीरिया व्हेन ट्रीटेड विद पोटेशियम आयोडाइड सॉल्यूशन एंड देन व्हेन वॉश्ड विद अल्कोहल, उसके बाद दोज बैक्टीरिया हु रिटेन्ड ब्लू कलर, दे आर कॉल्ड ग्राम पॉजिटिव, और जिनमें ब्लू कलर नहीं रहा, they are called gram negative bacteria. Bacteria can be motile or non-motile. If the bacteria, they are motile, they can move from one place to another. They have a thin filamentous extension on their cell wall, which is called flagella. अभी हमने देखा depending upon the arrangement of flagella, the bacteria, they have been divided into various categories, and besides flagella, the structure present which help in the movement in bacteria, they can be pili and fimbriae. These are pili are elongated tube-like structures which are made up of a special protein, and fimbriae, they are small bristle-like fibers which are present all over the cell. So this is the prokaryotic cell.